Okay, everyone, I think we're ready to start back for the afternoon session. I don't know about you all, but I sure am enjoying being back together. We're coming up with all kinds of good ideas for future work coming out of this conference. One that we came up with over lunch was maybe next year we have a panel discussion with current students and old professors to talk about their career trajectory. They really enjoyed Dr. DePerrio's career trajectory, because I don't know about you, but I had no idea this is what I'd end up doing for my career. And I think we have so many students who attend this, and maybe even invite some high school students in to see how people ended up where they are and the exciting, fun things they got to do in their lives for a job. Speaking of that, I don't know anybody who gets more excited about ticks and insects than this next speaker. She's one of my all-time favorite students I ever had as to serve on her committee and work with her. Paul Nora was another one. Paul, you were one of my favorites too. Anna Pasternak is a PhD candidate in the Department of Epidemiology at the University of Kentucky and a fellow in the Central Appalachian Regional Education Research Center. She received her bachelor's degree in biology and a certificate in environmental science from Ohio University in 2018. She went on to study holistic approaches for the control of insect pests and work as a mosquito control technician for the Athens County Health Department in Ohio. She joined UK in 2019, where she has been working with the Kentucky Department for Public Health to better understand tick and tick-borne pathogen dynamics in Kentucky by performing statewide surveillance. That's statewide. You were in every county? No, not quite. We have 120 counties in Kentucky, so that's a big job to cover Kentucky. She hopes that by uncovering what tick species are, where, what pathogens they are vectoring, and when they are active, can provide resources for public health officials, physicians, veterinarians, and everyone in between to increase their awareness about ticks and tick-borne diseases. Help me welcome Anna.
Alrighty, thank you, Dr. Sanderson, for that, and thank you all for staying around to listen. I always like when they put me after lunch because I can make the joke that now that we're all full as a tick, we can learn about ticks. So I'm going to be talking about surveillance for ticks and tick-borne pathogens here in Kentucky. So before we get started, how many of you have ever encountered a tick? How many of you enjoyed that experience? Good. Oh, one weirdo in the crowd, but that's okay. So ticks are rather unlikable. You know, they're vectors, they're parasites, and unfortunately, they're all over the world. So there are over 800 species of tick worldwide. They're found on all continents, and they can be grouped down into 3 families, the most important of which is the family of hard ticks, Ixotidae. So these are the #1 vector of disease to humans in the United States. They're #2 in the world, behind, of course, mosquitoes. All stages and species of tick are blood-feeding ectoparasites, so they're attaching to the outside of us and feeding on our blood. And it's during this time of feeding that they're able to pick up and then transmit pathogens that can go on to make us sick. So all ticks go through 4 stages of life, the first of which is a tiny, tiny egg. So eggs are laid in grassy areas or in leaf litter on the forest floor, and then females can lay up to thousands of eggs in a batch. Scarily enough, 23,000 eggs is the record. I don't want that many eggs. I'm sure neither of you do either. All of those eggs will then hatch into larvae. The larvae are very small. They're called seed ticks or turkey mites. They're about the size of a grain of sand. The good news is that they usually do not transmit disease. Most pathogens are picked up during the blood meal. Because larvae have not taken a blood meal, they're more or less just a nuisance. Once they take that blood meal, they'll molt into a nymph. Nymphs are a bit bigger, about the size of a poppy seed, but they're still really small. And because they have pathogens that they may have picked up during that last meal, they pose the greatest threat of transmission for disease-causing pathogens. Because they're easy to overlook, some pathogens can be transferred in only a few hours. Thankfully, others, like our first speaker noted, the bacteria for Lyme disease takes about 24 hours or over. The last stage is an adult. So adults are about the size of an apple seed, much larger, much easier to see than the immatures. Males do not need to feed, so females are going to be the only ones that are out looking for a meal during this stage. After she has her meal, she'll mate, lay eggs, die, and then the whole cycle will repeat over again. So ticks are very tough, they're very hardy, but their survival is restricted by some factors. And just like any other organism, certain conditions need to be met in order for a tick to survive and thrive. 3 major factors that influence tick survivability are humidity, temperature, and then the availability of food and shelter. Humidity is arguably the most important, so ticks, like a lot of other arthropods, cannot desiccate, they can't dry out. So they need damp microclimates in order to survive. This can look like rotting vegetation, so think of a fallen tree, or a damp leaf litter on the forest floor. Desiccation tolerance will vary by species, and this goes on to influence habitat preferences. So certain habitat preferences of a tick will then go on to influence whether or not a disease is prevalent in an area, because certain diseases are only transmitted by certain ticks. So if a tick species cannot survive in, let's say, the Southwest, for example, you don't see a lot of Lyme disease, Ixodes species don't have a high desiccation tolerance, so they're not able to survive. Another important factor is temperature. So activity generally will increase as temperature does, as long as humidity levels are adequate. There is some activity of tick species in colder temperatures, like Ixodes scapularis, which is the Lyme disease vector. Once conditions start to get a little unfavorable, ticks will enter diapause, which is like a sort of hibernation in order to survive, but they need shelter to do so. And that is why accessibility to food and shelter are the last factors. So ideally, no shelter equals no survival. Ticks need shelter not only for diapause, but for molting and for laying their eggs in. Ticks are also reliant solely on blood for nutrients. So anywhere that provides easy accessibility to hosts, whether that be free-ranging livestock, wildlife, or outdoor pets running around, that's going to be an ideal place for the ticks to live in. And ticks really like the East Coast, as you can see by these distribution maps here in yellow. And it's no surprise, really, because it's hot, it's humid. We have plenty of natural areas and plenty of wildlife or outdoor pets or livestock running around for them to feed on. And so because they are so medically important, then we like to conduct surveillance to figure out which areas in those distributions are at higher risk for tick exposure. Does it come down to a certain habitat type? Does it come down to, an occupation? And so that's what my dissertation work has been aimed at, is performing surveillance for ticks and then their associated pathogens in Kentucky. The primary objective is to determine the distribution, the abundance, and the seasonality of different tick vectors, and then look at the distribution and estimate the prevalence of major tick-borne pathogens in Kentucky. So in Kentucky, the 3 top diseases are spotted fever rickettsiosis, ehrlichiosis, and Lyme disease. And to make it simpler for myself, so I don't have to write up everything about ticks for my dissertation, these are the 3 chapters that I've been focusing in on with this surveillance. So to perform the surveillance, we've implemented 2 different collection methods. The first is active surveillance. So this, you might not be able to tell, but it is our fierce leader, Dr. Sanderson, out in the woods of western Kentucky, collecting ticks on this drag sheet that you wave around in the grass. The ticks will grab onto that, and then they're easy to collect from the drag. Passive surveillance has also been implemented in the form of a Kentucky Tick Submission Program. So this is a program that is restricted to individuals in Kentucky. If they find a tick, they can send it in, and then we include that data in our surveillance. So I'm going to go through for the rest of the talk and just kind of breeze through the main tick species that we've collected, kind of give over the distribution, the abundance, and the seasonality of them and their associated pathogens, to give you an idea of what you're going to see in Kentucky, but these are probably also going to be pretty common in most of the Southeast. So the first up is the Lone Star tick. So this is a pretty common tick. It's a very aggressive biter. The adults are easy to ID because they have brown legs, brown body, long mouth parts, and then females will have a white dot in the center of their back, and males will have smaller white dots along the bottom of their body. So Lone Star tick and Ehrlichia chafiensis, which is the agent for ehrlichiosis, is pretty widespread in Kentucky. So for the past 4 years, when I've been doing the surveillance, I've been able to collect Lone Stars in 116 out of 120 Kentucky counties. That's roughly 96% of the state. So there's just a few counties that appear to be safe from the Lone Star tick. But you can see that it's very widespread. Ehrlichiosis also appears, or the agent for ehrlichiosis also appears to be fairly widespread. Lone Star tick is by far the most abundant species that we have in Kentucky. So I've collected over 9 species throughout the surveillance, and you can see that Lone Star tick makes up 79%. Of that 79%, over half are larvae. So these are those seed ticks or those turkey mites. Lone Star ticks I'll show in a second, have like a bimodal or seasonality, and so the adults and the nymphs will come out earlier in the summer, and larvae will peak later in August. So you can see that here we have adults and nymphs in blue and red, and then yellow is the larvae. And so you can see that they may come out as early as March, but even, I don't know if you can see it, but there's a little collection in November if it's warm enough for larvae. So they have a pretty wide range in terms of when they're going to be active throughout the year. And then at least 2.2% are infected with the primary agent for ehrlichiosis. So the CDC designates rules for conducting surveillance, for testing for pathogens. Because host-collected ticks contain blood of the animal or the person, and when I test, I can't differentiate whether it was the tick that was infected with the pathogen or the host that was infected. When I calculate these percentages, I'm only using ticks that were collected from the field, so only ticks collected through that active surveillance. And at least 2.2% are carrying the ehrlichiosis agent. So we heard about ehrlichiosis this morning a little bit. I'll just give a quick overview again. So ehrlichiosis can be caused by Ehrlichia species bacteria. In humans, it's mostly caused by Ehrlichia chafiensis. It's primarily associated with transmission through Lone Star tick or amblyomma tick bites. And then deer and mice are common reservoirs in the environment. Flu-like symptoms are a great way to describe signs and symptoms of infection. And then 30% of cases, mostly in children or older people, will develop a small rash. Ehrlichiosis has really increased in the last couple of decades. So in the US, they have more than doubled. And then between 2010 and 2019, just in Kentucky, we had a 537% increase. Kentucky is also one of the highest incidence states nationwide for ehrlichiosis, so it is a big concern. Lone Star ticks are also associated with alpha-gal syndrome. So alpha-gal is, as we heard, more of an allergic reaction than anything else. Symptoms occur after eating red meat or exposure to products that also contain the sugar molecule. And then the notable thing is that symptoms will occur several hours after ingestion or exposure. This can be triggered by tick bites, and also people who maybe have this allergy and then seem to get a little bit better. If they're bit by another tick, then that can flare up their allergy again. Now there's growing evidence that Lone Star tick is the one that's primarily associated with alpha-gal, but it is not ruled out that other ticks cannot also irritate this. So with alpha-gal, prevention and management is very important. So you want to avoid tick bites, of course avoid products with red meat or anything that's going to contain that sugar molecule, and then also seek medical care if your symptoms get severe enough. Now moving on to the dog tick, the American dog tick specifically. This is also a pretty easy tick to ID as adults. So they have brown legs, brown bodies, this very complex white pattern on their back, and then very short mouth parts. The American dog tick is also pretty widespread in Kentucky. Again, its distribution is shown in gray here, but you can tell it's not quite as widely distributed as Lone Star. And then in specific relation to Rocky Mountain spotted fever, The pathogen Rickettsia rickettsii that causes that, I've only detected it in 3 counties. So Rickettsia rickettsii, even though it is a very severe disease, is very often not detected in American dog ticks. There's different theories that go into why this is, but it has been detected in these 3 counties through my surveillance. It's important to note that the ticks that harbored this pathogen were blood-fed, and so because of that, I can't definitively say that the ticks were infected. It may be that the host was infected, but with our passive surveillance system, we do ask for travel history within the last 2 weeks. So we heard about how important travel history is with mosquito-borne diseases. We apply the same framework here. If the pet or the person, whoever the tick came from, had not traveled outside the county in the previous 2 weeks, then we have some confidence that they picked up the pathogen in that area. American dog tick is the 2nd most abundant species in Kentucky, even though it only makes up 12% of the whole population. And then of that 12%, most were adults, which is not uncommon. So American dog tick immatures will feed on small mammals. They're not very often encountered by humans, and they're not very often picked up in surveillance. So adults are going to be the one that's going to cause the biggest problem. And then they are active, overlapping with Lone Star tick, but they seem to peak in May and June, and then seem to not die out, but go into that diapause much earlier than the Lone Stars do. I mentioned this already, so very few of the American dog ticks came back positive with the Rocky Mountain spotted fever agent, and then the ones that did had come from were collected from hosts, so we cannot definitively say that the ticks were the ones infected. It may have been that the host was infected, but we can be sure to say that the Rocky Mountain spotted fever agent, Rickettsia rickettsii, is in Kentucky and likely in those counties. So spotted fever rickettsiosis is an umbrella term for a bunch of different Rickettsia species bacterial infections, Rickettsia rickettsii being the most notable. So Rocky Mountain spotted fever is primarily transmitted by the tick Dermacentor variabilis, the American dog tick, but we heard that transmission by other species is possible in other regions. Symptoms are similar with ehrlichiosis, where they're flu-like. There is this characteristic red spotted rash that can appear, and then you also often have fever, and then an eschar at the bite site. So spotted fever rickettsiosis, specifically Rocky Mountain, is very severe. 20 to 25% of untreated cases will be fatal by the 9th day. US cases have doubled. This is specifically for spotted fever rickettsiosis in the last decade. And then between 2010 and 2019 in Kentucky, cases increased over 5,000%. I do not do the disease surveillance. I don't know the details of that, but that is a huge increase. Kentucky is also one of the highest incidence states for this disease nationwide. And then moving on to everyone's favorite, the black-legged tick. So black-legged tick, Ixodes scapularis, are very unique when compared to other tick species. So they have black legs, a black scutum. Females have longer mouth parts, whereas males appear a little shorter. And then females will have a red or orange kind of underbelly. Black-legged ticks are not nearly as common or as widely distributed as the other 2 that I've mentioned, but they are present in Kentucky, which 10 years ago was something quite a few people would argue against. The black dots represent detections of Borrelia burgdorferi, so you can see that we've also detected that in our tick populations, but again, it is not nearly as widespread as what I've already talked on. And then it's our 3rd most common species, so it makes up 7% of the tick population. Most of the ticks collected were adults. Nymphs and larvae can be a little trickier to catch, especially as you get out of that northeast region and into the southern, but we were able to collect some, and from those we can say that they're active all the time. So tick season is all the time, at least in Kentucky, but likely in other states. You see the nymphs and the larvae active, May to about August, and then the adults will emerge in October and remain active all the way through May if temperatures allow. With black-legged ticks, we did not have nearly as large of a sample size. So with Lone Star ticks, I've collected a little over 8,000 of them. With American dog ticks, it's almost 2,000, but only 674 black-legged ticks have been collected. So in those 674, of the ones collected in the environment, it's closer to about 200. And then 4.6% of those were detected or had Borrelia burgdorferi detected in them. And Lyme disease is, of course, caused by Borrelia burgdorferi bacteria. There are cases caused by Borrelia mayoni in the Midwest, but not in Kentucky or the larger Southeast, as far as I'm aware. Lyme disease is specifically transmitted through bites of Ixodes ticks, so you're not going to get this with Lone Star ticks or American dog ticks. And then the symptoms are, again, flu-like, but with specific fatigue and stiffness in your joints, and then the EM, the bullseye rash that we heard about this morning. So the most common tick-borne disease in the US is Lyme disease. It's considered an emerging epidemic. You can see by this little moving map that it's really starting to increase and creep in out of that northeastern region. Between 2010 and 2019 in Kentucky, cases also increased by 340%. I do want to note that that increase went from 5 cases in 2010 to 22 in 2019. So there is still not a ton, but we are seeing a gradual increase. And then another important note with Lyme disease is that without early treatment, without proper treatment, it can evolve into post-treatment Lyme disease syndrome. Some people argue that this is also considered chronic Lyme. So I know that was kind of a quick talk. I was told 20 minutes. So here's my summary slide. Ticks are the #1 vector of disease to humans in the US, but nymphs and adults are more likely to transmit pathogens than the larvae. So if you're out, if you're hunting, if you're working and you kneel down, stand up and you're absolutely covered in those little seed ticks, the primary concern is that they're just a nuisance. Ticks thrive in warm, humid, and vegetative environments that also provide easy accessibility to hosts. So by creating unfriendly habitats and limiting accessibility to those hosts can help in controlling ticks. And then Lone Star, American dog, and black-legged ticks are the most common ones that we see here in Kentucky. And between these, tick season is every season. So you should always be on the lookout for ticks when you're coming back in from working outdoors. You should always use your DEET, always use your permethrin, especially if you're going to be spending a lot of time in those woodsy habitats. And then we're after lunch, so I can show this next slide. You should always be very careful and check for ticks thoroughly. This picture on the left is a tick in a boy's ear canal up in the New England area. And then the right actually happened in Kentucky. It was a lineman who had been to work. He got home, had something in his eye. He couldn't get it out, went to the eye doctor, and there was a tick larvae that had attached to his eyeball. So be very careful. And then that is what I have. I know that was kind of quick, but I'll be here, and if anyone has any questions, I can answer those.
First, I just want to point out that all your top-level bullets were ticks, which is like peak presentation humor. My follow-up was, so you said that the Lone Star ticks are like kind of aggressive, and we've also noticed that like roughly 80% of the ticks you've collected are the Lone Star ticks. Do you think that it's maybe just because they're aggressive and they might naturally be collected more easily? And if that's the case, does it really matter, since then they're likely the ones that are going to attach to people anyway?
So Lone Star ticks, they are aggressive. They have been documented to actually chase after a host. So most ticks are fairly lazy. They like you to come to them. But Lone Star ticks, once they detect CO2, body heat, whatever it is that gives you away, they are known to chase several meters after you. I've had this happen to me in the field. So I do think that provides kind of a bias when you're doing active collections. But I think when you look at it overall, they're still the most common species by far. I always argue that Lone Star ticks are the ones we need to be the most concerned about. There's a long list of diseases they're associated with. They're aggressive biters. They have a pretty long seasonality. And so I think, yes, there is some bias in collecting, but I don't think that means that we should be less concerned about them.
Thank you very much for your presentation. In the interest of research to practice, will you care on going a little bit more detail in terms of, what can we do about this, public policies, Thank you.
Yeah, so like tick control, is that what you're getting at? Okay, yeah, so there are, unfortunately, tick control is not universal. Because different species have different habitat preferences, they have different host preferences, and they're a little tricky to control because they're easily moved around by hosts. It's not as, simple when you're looking at, like a cockroach infestation or something like that. So with most situations, land maintenance is a really important control measure. So keeping grass cut, removing leaf litter, kind of creating unfriendly tick habitat is going to be your best bet. Unfortunately, that doesn't always apply very well in agriculture or forestry because you're not wanting to mow down 150 acres every week, and you can't necessarily apply pesticides to grass that maybe your cattle need to feed on. So once you get away from kind of your common practices, there are other options like using acaricides for livestock. If you work in a park system, trying to maintain your trail very well to keep ticks from latching onto people as they brush up against the tall grass right there. And then there are other kind of commercial options like tick tubes, which you can buy on Amazon. It's cotton that's treated with an acaricide, and then you can throw that out in your lawn, and little rodents and birds will pick up the cotton and put it in their nests, and then they have a treated nest. Yeah, very cool idea. There's also a tick robot from, I think it's a university out of North Carolina. You can't buy it, but it is a very cool research, advance.
Hi, Anna. Great presentation.
Thanks, Kara.
I was wondering if you're going to look at the data or if you already have looked at the data in any spatial way other than county, like maybe ecoregion or terrain-based, just because counties are made by humans, so ticks don't really know oh, I'm going to stay in this county or something,
I have not, so all the data that I collect, because I'm funded by the State Health Department, goes to them at the end of the year, and then it gets uploaded to ArboNet, which is the CDC's database. They run on a county level scale, and so that's what I've run on. But I do know at UK, especially in forestry department, there are some, I think Maria Topmiller, she's looking at land cover use in relation to Lyme disease and different things like that. So I'm not doing that, but there is research on it.
That was a great question, Kara, because it makes a point here is, here we've got Anna as an entomologist focused on occupational health issues, and Maria is a forestry major also focused on occupational health issues. And just to tell you a little bit more about her study, she was trying to get down to the level of forest cover versus Lyme disease incidence. And what she was finding was in the counties that are almost 100% forest cover, you don't see much Lyme disease. And this is even looking at rates because there's fewer people too, right? In urban areas, you don't have very much Lyme disease. You almost have to have a 40 to 60% blend because where do the ticks come from? They've got to eat something, right? They've got to bite something like a deer. Well, the deer has to have something to eat. If it's only forest land, they kind of need a little bit of a crop and they need a little bit of forest land. So how does that help us? If we're going to target our education programs and target our interventions, and this is one way to go. But I mean, again, you're setting us up well for the workshop this afternoon, but Did you analyze for the ticks for what they had inside them? Did you do that in your lab?
Yeah, I did it.
How
The-
common is that to have those kinds of facilities? Is that pretty easy to do, or?
I think it depends on the relationships that you have. So I know that right now, this is the first statewide effort that Kentucky's ever had for tick surveillance. They are having me do it as a graduate student because they did not have the resources or the funding to have their state lab do it, or I don't even know if they have a state lab actually, to do the testing for them. Other states like Virginia and Tennessee, they do some testing in their state lab, but it kind of just varies based off of, I think, state funding. I'm in a lab that is well supported by grants, so I don't know if it's easily accessible or not. I think it kind of comes down to your relationship with academic partners. And
Is that PCR testing? Is that what you use?
yeah, qPCR testing.
Thank you, Anna, for your presentation. So I grew up in Tennessee, and my family had a logging business. We owned a farm, and so I was always out in the woods cutting trees. And as I'm sure, being out in rural Kentucky in the forested lands, we wear blue jeans, steel-toed boots, and a shirt, and carrying chainsaw and wedges in our pockets. And so, you know, one of the issues in being out with logging and being out in the forest is, you know, if I'm out hot and sweaty and it's a 90-degree day with 90% humidity, you know, I'm not wanting to spray, you know, repellents on and then trying to carry that around. And then the other issue, you know, getting home and just even checking for ticks and checking for nymphs, right? Because that's not in the back of my mind. I'm trying not to die from having a tree fall on me or get hurt, right? And wearing a hard hat. So I wondered about the sense of, you know, for forestry workers or loggers or people who are out in the forestry, you know, how frequently were they checking themselves for ticks at the end of the day? Because, you know, when I was doing this in the early 90s, it was finding the tick on the back of my leg, you know, a couple days later, right? And so I became a little bit more sensitive to that as I got my nursing degree and took care of people who had some of these vector-borne illnesses. But what was your sense in terms of persons using PPE? Because we can't eliminate forestry, right? You know, cover when we're out cutting, but sense of PPE in terms of repellents, and then just checking themselves for ticks after they've been out cutting or working in the forest.
So I haven't looked specifically at what people are or not doing in terms of tick control and PPE, but I spoke at the Kentucky Forestry Conference a couple of weeks ago, and a lot of them came up with the same concerns. They're like, it's hot, I have other concerns, you know, I don't want to be spraying sticky DEET on me, and I always recommend treating your clothes. So I get a can of permethrin from Walmart, I treat my clothes. It's good for several washes, and I've had a handful of tick bites in the 4.5 years that I've been doing this. So when you use it appropriately, it is very effective. There are other little things that you can do, like when you have on long pants, tucking those into your boots, pulling your socks up over. People who hunt will take duct tape inside out and wrap it around their legs so the ticks get stuck as they try to climb up the legs. So I think it really comes down to what an individual or what a group is willing to do and the amount of effort they're willing to put into it. I don't like to wear, I'll be honest, I don't like to wear DEET or bug spray. It's sticky, it gets on my hands, and then no matter how many times I wash, I go to eat my sandwich for lunch and I can still taste it. So I really like permethrin. I think the biggest thing is just remembering to do tick checks. Something that I've been involved in with the different county agents is trying to get them to put up signs along hiking trails or different things to just remind yourself, stop, take a quick look for ticks. If I could take everybody out in the field to show you how small these ticks are, how easily they get on you, I think that people would have a much better mindset to this is very important, I do need to check. But until then, I think the best thing that we can do is educate, provide realistic approaches for them that work for them and are not going to be just another chore while they're trying to do their job.
you. Thank you, Anna. So we've set aside the rest of the day for some brainstorming and workshop opportunities here. This is something we've been doing here at SouthON now for the last few years. I guess we didn't get the opportunity to do it quite so much when we were remote, but this gives us an opportunity to explore some questions and how we might work together. Whether you actually have a passion for doing this kind of work or not, you might certainly contribute to ideas that others might take upon. I'm open to questions and also to how we might structure the workshop, whether we could do it all together, we're not a big group, or whether we want to separate into groups. But one area that I thought about is the question is, what occupations are at greatest risk? And this is an important question because it helps target our intervention and our education efforts, right? And what's key to that is we see that there's lots of problems with our surveillance. How can we improve that? I think that's an open question for us as a group, occupational safety and health professionals. How can we intervene to help convince CDC and other people that occupation and industry are important factors to collect, and where it is collected, in some groups, how can we gather that data and use it to guide our work? That was question number one. Question number two is then, Anna just ended her talk with, boy, I wish I could take everybody in the field and show them how small these ticks are and how easy they are to get on you, and that the diseases are here. Our surveillance, such as it is showing that. How do we create a greater awareness? What kinds of educational tools? Who should we target that for? How will we implement that? The next question I had is, well, what research gaps? For example, evaluating these protective measures, coming up with how do we have laboratories that can actually analyze for these things and be vigilant for new and emerging diseases, which comes to the point of maybe building capacity for this work. So also, our center has, our centers, we have 2 centers that have pilot grant programs each year. I know many of you are from states and may be interested in having some pilot grant effort money to do this kind of work. Each year, we still take a broad range of occupational topics that people may want to do, but we also have a focus. And this year we've chosen infectious vector-borne diseases. So if you would like to apply to our center, we have all your addresses. We'll send out the notice when it comes out in April for the ERC and in July for the Ag Center. As long as it has an occupational hook, it's something that we can fund. So if you said, hey, we want to try training this group and we want to develop some materials and there needs to be an evaluation component to it, we would like to do that, or whatever topic you come up with, we're willing to put some funding on the table to provide to people as well. So how would you like to run the workshop? You want to divide up into about 3 groups? Think that's a good idea? Okay, let's try that. Let's see if we can't divide into maybe, help me here, see, how about a group that talks about surveillance? Maybe we put them over there. We have a group that talks about education and awareness back there. A group that talks about research gaps. Do we need a 4th group, or is 3 about enough to get started? 3 is good? All right, surveillance, education and training, awareness, and research gaps. And you're welcome to take a break too if you want, and go to the bathroom. I think there's coffee and such, and then we kind of gather, and we'll do this for approximately an hour, and then get back together and see where we're at. Okay, you all, we're going to get back together now. Hopefully, you know a whole lot more about vector-borne diseases now than you did this morning. This is kind of my area, and I'd have to say I know a whole lot more about vector-borne diseases than I did this morning. So we have 4 groups, and what we would like to do is you come up, have at least one person, you can have more if you want, but at least one person come up, and we're recording this. I think it's also being streamed out there, but this way we'll capture some of the great ideas that are coming out of the groups that we can take back home with us and act upon, make some changes. I've already heard several good ideas I want to steal. And if, like we said, if you need funding for this, we'll be putting pilot grant announcements out. Dr. Bernard said he would even chime in some funds and put some money on the table for folks to work on projects such as this. So I'm going to turn to the group on the extreme right to go first. And then we'll just take whatever time. Oh, Ashley wanted me to make some announcements. Let's see. Announcement number 1 is the shuttle bus just started and it runs till 4:30. Is that right? Okay. What else did you tell me to say? What's that?
The shuttle bus will be leaving from Embassy Suites for the Busch Gardens tour for those of pre-registered. I believe the first one might leave at 7:15, just in case we need to make 2 rounds, but we definitely need to be there before 8 a.m. They are giving us a tour before the park usually opens at 10 a.m., so our tour is 8 a.m. to 10 a.m., and they have to specifically let us in. So anyone that's doing the tour, definitely be there at the front gates before 8 a.m. I have emailed out instructions, driving instructions and parking instructions to almost everyone, those that registered just recently. I'll be sending out an email to you all as well.
Okay. Thank you. All right.
Oh, no. Guys, it's still.
Our group was bad about writing things down, but we talked about a lot of different ideas, and so we're going to try to share them. So we talked about the possibility of looking at different sources of claims data that already exist, like insurance claims data or workers' claims data, whether that be, of course, there's issues potentially with workers' claims data because of not knowing necessarily where it's related or not. So maybe possibly looking at denied claims as well for workers' compensation claims data. We talked about a project at University of Florida where they're trying to work with a bunch of different hospital systems that all use Epic to try to get them to collect industry and occupation and get that included, and also some of the challenges around that with all these different EHRs, and also different hospitals get to sort of choose what is included on their electronic health record. Let me see, what else did we talk about? Did you get some other ones?
Yes. So one of the other things that we were focusing on sort of as a broader topic was the lack of standardization from the data as it sort of flows in. One of the things that we were looking at was also record sharing from different departments. Specifically, the military healthcare system was brought up as a good example of being able to pull data from various sites around the country, and it will still come in. But having more of a mandate across the different spheres of healthcare to report some of these illnesses, these vectors of transmission. One other sort of dovetailed into that was the idea of animals and looking at veterinarians and some of the zoonotic vectors of transmission, just to really understand where these are coming from and where they're going, because as sort of was touched on in some of the other presentations, with all the various changing factors around the world, there will be different sort of disease in different places, and we need to be able to track that.
We also talked a little bit about working with like local health systems, and like the National Association of City and County Health Officials has like a vector-borne mentorship program. And so working with those like health departments, working with NACCHO and the health departments potentially that they have already identified that work on vector-borne, and, you know, talking with them about the possibility of collecting like industry and occupation when they're doing surveillance. So I think those are.
Yeah, that was about it.
Did we miss anything, guys? Okay.
Thank you very much. Veterinarians, if a dog gets rabies, it doesn't go to notifiable diseases, does it? It's only humans. Is that right? If we have a rabid animal, does that get reported? It does? It does, okay. All right. It does. Okay. All right. Next group.
Hello, we are from representing education portion, and we focus mainly on ticks. So for tick education, we decided that we could have QR codes at trails with tick info. And in the QR code, it'll tell you what to look for and show you how to remove it, a little video. You could also have stores and hunting and hiker brands put it in their campaign for tick removal. And at their checkout counters, they could have a free gift, which would include a magnifying glass, tweezers, and a QR code for proper removal of the tick. And those same little free gifts could be given to high school kids or future farmers of America, so that they can teach people about tick removal, but more importantly, go to the elementary schools. So that will be high schoolers teaching elementary school kids how to look for a tick and how to remove a tick, but for them to tell their parents when they should check them for ticks after they're outside. So another thing is giving education through tourism. So people at the airports and train stations, you could see a sign that says, hey, you know Kentucky for the Derby, but also know that we have ticks. So this is how you check for ticks and remove them. Also to know that have primary care practitioners talk about tick prevention and removal, vets to discuss tick prevention and removal for the animals and the owners, and also promethazine is toxic to cats, so be mindful of that. And then last but not least, so if you want to have a really good campaign, people you should have it affect their heart, right? So you can have a little video of a person walking on a trail and a tick come up and bite them, and at the end of this trail they have a burger, and then they take a bite of the burger and they get sick, because people are more concerned about getting sick and not being able to eat a burger than they are about getting a tick bite. So that could be the commercial with big graphics and over dramatization, and then people will be more mindful to look out for ticks. That's it. Thank you.
Nice job. Next group.
So I gather from Dr. Sanders' orders, we were to do open research questions, so it does relate to some of these other ones as well as some other topics. So the first area was we started off with was just thinking about how to get employee buy-in, which relates to the education side, but also how to get management buy-in to any vector-borne disease prevention efforts, particularly if you can't get management buy-in on other things that we've known about for a long time, like silicosis and other things. So how could you get them to also adopt a vector-borne based intervention? Ensuring that the educational interventions may be the most culturally competent, because we know there's a lot of disparity in determining and diversity in the workforce that's at greatest risk. A lot about identification of barriers to implementation. Made that notes on that. It kept coming up multiple times. One of the potential barriers also goes back to the slide we saw earlier, the very beginning, about how to improve the classification of work-related illnesses as well as work-related mortality. What happens if somebody develops one of these illnesses at work and then has to live with the arthritis for the rest of their life? They've no other history, but their workplace history is very specific to something that would have put them at increased risk. What are the management implications of developing or implementing any interventions? And then a lot on anonymous surveys or focus groups of actual workers regarding interventions that could be developed, because we know that surveys of managers versus employees and the settings in which those surveys are done can change the outcomes. Let's see, a lot of questions related to how do we obtain the existing knowledge, attitudes, and perceptions of employers? We should presumably do those before we do rolling out of any interventions. What are the employee perceptions of, or the potential user perceptions of DEET, permethrin, eucalyptus oil, lemongrass, and all the other ones, versus other preventive measures that could be more aggressive, like we joked about wrapping your legs with duct tape or something? What are the existing tox studies or more modern tox studies on not just a little bit of permethrin exposure, but chronic exposure to permethrin-treated pans? We're not talking, 10-year, 20-year, 30-year of doing this, if this is going to be something. Are those studies out there? If they are, how do we incorporate them into educational designs? Are there biomarker-based studies that exist on these different treatment methods that potentially could allay concerns or maybe confirm some concerns that workers may have? And then in regards to other populations that may be beneficial as a proxy rather than going directly to the workforce, using state ag type programs like fairgrounds, county extension agents. A lot of outdoor workers also tend to be more likely to hunt and fish, so using hunting and fishing registries and studies on hunters and fishermen as a means for assessing intervention, risk for success at least. Other than that, last one's kind of a surveillance-related question. I'm from Kentucky, so the president of the National Association of Biological Field Stations is at Thomas More College, and they're always looking for ways to study ecological things. And we started talking about Robinson Forest as an old-growth forest that UK operates in southeast Kentucky. EKU's got Lily Corner Woods. There's these field stations, these academic ones that are looking for things to do. Their workers at these field stations may be, good folks to do some knowledge, assessments relating these conditions, as well as possibly helping support surveillance across the country using existing field stations with a lot of existing data, and they know their area well, and the mosquitoes and things don't realize that they're at a field station or not. So, so that was it. We had a pretty good list. It was a great group to work with. Thank you.
Great job. Next group.
You can tell that I work with Rebecca because our first two things were exactly the same as what she said. So we had standardizing data collection across both borders, so different regions, and time, so like handoff of data collection programs from one researcher to anyone that follows after them. And then we also went with workers' comp as a source of information, but we were spinning that as sort of a way to look at the health economy, cost of disease, so talk with economists and find a way to make prevention the more financially attractive outcome than having a worker get sick and then having to retrain and replace and all that stuff. Another thing we had was to educate providers on like how serious the outcomes of vector-borne illnesses can be, and that these are actual threats in our community. I said, like, growing up, I thought of Rocky Mountain spotted fever as like the out west disease. Like, that doesn't happen where the Appalachian Mountains are. So I'm sure there are plenty of physicians that think that too. And we need to educate the community, but then we were thinking about how there can be dangers of having some information which can lead to misinformation when someone thinks they know something about a vector-borne disease and they're telling all their friends, and that's not necessarily the truth. And then some like small outreach things we had thought about was pointing out how small ticks are by maybe embedding a few in a block of acrylic or epoxy at a trailhead to give people an idea of like, this is how small you need to be looking at. And then the duct tape idea, people talking about duct tape, I thought you could also just put duct tape at trailheads, because that's a pretty cost-effective intervention, a few dollars at a time. And just a note about how you can wrap your pant legs in duct tape and prevent tick bites. And then one last thing is finding smarter ways to map and analyze data, so spinning off sort of Maria's land cover project.
Did I leave anybody out? No? Well, Tom, I think your idea of a workshop some 6 or 7 years ago turned out to be a great idea to not only make SouthON educational, but interventionist, so that I think we captured some really good ideas today. So don't be surprised if one of these days you go to a trailhead and there's some in a block, all these ticks that you should be looking at, and a roll of duct tape or what have you. So some of these ideas are likely to come to fruition. And we're always looking for other good ideas. We have SouthON every year. We've still got at least another year of funding for it from NIOSH. And we're well represented by the centers, the NIOSH centers here. So if you have a topic idea, share those with your centers. They get together. In fact, Ashley told me she was already making notes, and I'm sure she'll be pestering Kathy DeSilva and her other colleagues about what we're going to do next year. They'll start planning. It's like Macy's Day Parade. They'll start planning as soon as they get back home. So if there's ideas that you have for the Lexington meeting, certainly share it with those folks, and we'll see what we can do about presenting another topic of interest to you. But I thought this was a great one. I really learned a lot and enjoyed working with you all today. Lots more to come, a couple more days of really interesting presentations and talks. You see we've got a variety of posters already up. There'll be even more showing up tomorrow. So I hope everyone has a good evening. Enjoy your evening and networking with each other. Thanks.